الحمد لله والصلاة والسلام على أشرف خلق الله محمد بن عبد الله وعلى آله وصحبه ومن والاه، أما بعد، فحياكم الله وبياكم في المحاضرة الثانية لقسم علوم البحار، ونتشرف باستضافة الدكتور الزائر البروفيسور توماس بيانكي. طبعا يوم الاثنين ألقى محاضرة عن الكربون ودورته في البيئة البحرية، والآن اليوم راح يتكلم عن موضوع شيق يتعلق بالبايو ماركرز. طبعا اليوم اعتقد بعض طلبتنا درسوا البايو ماركرز، خاصة اللي درسوا معي ابلايد، مادة ابلايد درسوا البايو ماركرز او مارين كيمستري كورسز. اليوم دكتور بيانكي راح يتحفنا اكثر واكثر على انواع البايو ماركرز. طبعا Professor Bianchi, he's a full professor and holder of the John and Beverly Thompson Endwood Chair in the Geological Science at the University of Florida. Bianchi received his master's degree in ecology and evolution at the Stony Brook University in New York, and his PhD in marine science at the University of Maryland. He published over than 290 articles, top journals, Science, Nature, and he's still active and still writing papers. I think soon you're going to publish a new one in Nature about mud. There's a new paper about mud in Nature. Also, he's the sole or a co-author of 9 books, and these books, as a reference for the marine science students and whoever want to learn about the marine science, especially the biogeochemical cycle, the chemistry of the ocean. You must go through at least 1 or 2 of these books. I don't want to say all of them, which you should, I think, but at least 1 or 2. And he told us Monday he's also writing the 10th book about mud, which will be published soon, let's say in a year or maybe less, we'll see. So please welcome Professor Thomas Bianchi.
Okay, good afternoon, everyone. Thanks, Mohammed, for that very welcoming talk and introduction. So I'm very pleased to be here again. As I said the other day, each day that goes by, I keep learning more and more wonderful things about the university and this region in general. So today I'm going to right. Thank you. It'll be a shorter talk, and it will be a little more focused than the broader carbon cycle. And it's about something that I'm very interested in, have been using for many years, as Mohammed said, and that is with biomarkers. And so I'll talk again just a slide or two, just to get everyone who may not have been here the other day a little bit about the carbon cycle, look at the history of biomarkers, and where we get to think about which ones we need to use or can use for certain questions, and the limitations of using these particular molecules. Okay, so again, this is something many of you already have thought about, but if you start looking again at the flux of material from land to the coastal ocean, there are different pools. You have, of course, this atmospheric exchange that's going on with vascular plants and phytoplankton. You can see some of the long-term pools for tens of thousands of years to millions of years in terms of where carbon gets stored. So there's the long-term carbon cycle and there's the short-term carbon cycle, this being the long-term. The short-term is happening very quickly, where you have nutrients being taken up, ocean DIC going into DOC and POC. So this is all just sort of basic ecology, basic biological oceanography. once we start thinking about organic matter, right? We talked about terrestrial organic matter, there's marine organic matter. One of the other really important things when we think about what we're looking at is actually the size of the material and how oceanographers separate into pools of dissolved and particulate. So if you look here, you'll see this is the size in microns, nanometers, millimeters. Right around here is where you get the separation of particulate to dissolved, right? So dissolved is over here, and the particulate is over here, okay? And this is determined basically by what filter size you use. So most filters, if you take a seawater sample and you filter it through a 0.45 micron filter, what stays on the filter is POC, what goes through is dissolved organic carbon. These are just fundamental things that are part of the whole field of oceanography. That's the way we look at the world. We separate things into particles and dissolved constituents. So look at some of the things that are on both sides. Dissolved organic carbon, POC, zooplankton, phytoplankton bacteria. Viruses and macromolecules are on the dissolved side here. And you can see this is the dissolved organic carbon. Colloidal is high molecular weight dissolved organic carbon. You see 0.45 is used here, but sometimes we use 0.2 microns. So there's no real limitation to whether you can use 0.45 or 0.2. It depends on the question you're asking. So you will see it being used in both ways to separate POC and DOC in the literature. And there's a reason for that, but it's too long to talk about. And this is another aspect of it. In terms of the way we can separate things, we can use flow cytometers, sieves, filters, and ultrafilters in separating these. So it depends on how detailed you want to be in separating. So many of you have seen this and may have actually done this before. You can use typical vacuum pumps to filter water, and you get a nice white filter that will look like this after you've filtered through the water, and that's showing particles of sediment, it's showing particles of phytoplankton. You can do the same thing with the dissolved organic matter. Now I just confuse you more because now there's another size here, 0.7 microns. So 0.45, 0.2, or 0.7. It depends on the kind of filter, and again, I'll be happy to chat with you later if you want to talk about why use one versus the other. But you can see this is just basically collected from different terrestrial plants, leachate. If you look at the coastal ocean around marshes, you'll see brown water like this. Some of that is basically from the organic material that's coming out of the plants. Some of it's actually from mud particles, right? So it depends on what's causing that. And basically, you can see here, this is phytoplankton blooms. And so these are areas that are of particular interest, whether it's related to eutrophication or whether it's related to harmful algal blooms, but these are very common techniques to use. So some of the other methods that we have are sediment traps that we set up in the global ocean to trap particles that are sinking. It's very difficult to do this. It takes a lot of energy and time. But you can get a rate for how quickly particles are sinking in the water column, which is very important if you're doing chemistry of that. Water samples using CTD. I know some of you are familiar with this. That's sort of general oceanography sampling. Sediment cores. Mohammed knows this well. He works with coring. There are lots of various types of core devices, box cores, cast-in cores, piston cores. So it depends on what you're interested in. Are you interested in looking at the particles in the water? Are you interested in looking at what makes it to the bottom of the ocean or the bottom of the coastal ocean? And then we can then start looking at where they're from in biomarkers. This is just a little bit more. This is the box core and separating this. So again, I know some of you have already experienced some of this working with scientists here. So a little bit more before we get to the biomarkers. Some of the key questions we're interested in, you have DOM and POM coming in from land, You have DOM and POM coming in from the deep ocean. And then let's just say this is the coastal ocean here. So you're in between these two end members. Even though this says coastal ocean, this is sort of out deeper in the water. This would be sort of in between the land and the further offshore. And I won't get into all of this, but the point is that these are all the types of particles, the dissolved organic matter, small particles, fecal pellets, dead organisms that are sinking. All of this is very difficult to identify once it's lost its integrity of what it looks like. You can look under a microscope and see that it's phytoplankton, but once this material has been in the ocean for a while, the microbes decompose a lot of the structural components, and you can't really identify what it looks like any longer if you look in sediment. So you can't, it's not like a nice, clear phytoplankton pool that you're looking at. And so once we get down into the deeper parts of the ocean and the sediments, it's not recognizable anymore. And the other aspect of this is where it gets preserved. And I talked about this a lot. I just have one slide on it today. But depending upon whether it's an oxic system or an anoxic system, you can get good preservation of all of this organic matter that's sinking or coming in from rivers, or you can get poor preservation due to the presence of oxygen. So again, those are based on redox reactions, the efficiency at which microbes can respire under oxic versus anoxic conditions. So just, again, giving you some idea. Planktonic interreginous organic matter you get in the coastal ocean is highly mixed. And I had this slide the other day as well. And this is showing you where there are particular hotspots for burial and where we've used biomarkers. So that's just sort of a quick background on the size of particles, whether they're dissolved, particulate, how they get transported, where there are hotspots for production of organic matter, and then once we get unrecognizable material dissolved in particulate, how do we say where that's from? How do we know it's from a phytoplankton cell? How do we know it's from terrestrial material? Okay, so now the biomarkers. What is a biomarker? Basically, molecular marker or geochemical fossils, some people refer to them, but a molecule whose carbon skeleton can unambiguously be linked to that of a known biological precursor compound is perhaps one way to think about it. More generally, these are organic compounds found in sediments and the water column that have properties that can be directly related to known biological precursors. So if we don't have the structure, we don't see the diatom, we don't see the whatever dinoflagellate, how do we know how much of that carbon that's making it to the sediments and getting stored is from land or from the ocean? And you might say, why do we care? We want the ocean to continue preserving organic matter, and it helps us tremendously in sequestering this material away. And if we don't really know if it's coming from land, from rivers, or from runoff, or from coastal phytoplankton, we really can't manage the coastal ocean. We can't say we need to have less nutrients because we're seeing more phytoplankton make it to the bottom and cause hypoxia. Or if we put a dam upstream and now the organic matter and the particles coming into the ocean are blocked and they're being stored upriver, what does that mean to the coastal environment? So that's the reason we want to know something about sources. Now, this is the beginning of chromatography, and you could use this slide in a pure chemistry course. This is Svet, who is a Russian-Italian scientist. He was the first to use the term chromatography back in 1903. He did this very simply. If you've seen chalkboards where you have calcium carbonate, white powder, he obviously, this is going back a long time, the technology was not there. He made his own column, he packed this powder, and then he took basically some leaves exposed it to ether or alcohol, and then you get this nice green solution. And he poured the solution through this little column he made with this packed white material, calcium carbonate, and he realized that the compounds separated out. Let me just see. Yeah. So what they did was they separated out. So you got these various bands, and that chromatography is the science of separation. You'll hear gas chromatography, liquid chromatography. All it is taking a complex mixture of everything in solution or whatever, and trying to figure out what compounds are in there. So we use this in medical sciences, all of these things. Now, chromatography is a huge component of chemistry. So he was the first to really use this and give it a name, Svet. And then we started to look at what was there. And this is now the beginning of organic geochemistry. And basically, this is connecting the long-term carbon cycle of fossil fuels with where they came from. Basically, where does oil come from? Where does natural gas come from? We didn't really understand that until we started to be able to separate the compounds. So what you're looking at right here is a chlorophyll molecule. This is chlorophyll. It has a central tetrapyrrole structure with a central magnesium. It has a phytol chain, and basically you can take this molecule and start cutting it into pieces and decomposing it over time. So if you take a molecule of chlorophyll A and you put it in ocean sediments, it will decompose and get buried over time. And in its early burial, it can be converted to this phytol chain can be, phytol chain, yes, can be converted into phytane or pristane. Don't worry about that. And you can also get changes in whether there's magnesium or nickel in the center. So these are so we call diagenetic changes, changes to that molecule that was in the organism, that was in the diatoms, in the phytoplankton, that goes through further changes. That led to another discovery. This is Alfred Tribs, a German organic chemist. He was the father of organic geochemistry. Organic geochemistry is really understanding that these molecules of chlorophyll are linked to where you make oil and gas and so on. We didn't really understand where the fossil fuels came from. So once we could separate the molecules and we said, boy, there's a lot of this chlorophyll A remnant material in the oil and something like that, you could see that that was the connection. Okay, so that's again another early inception. This is essentially what's happening. Diagenesis, you have living organisms, lignin, carbohydrates, protein. As you go down in time, this goes from early diagenesis, which can be hundreds of years, decades. Now you start getting into catagenesis and metagenesis. So as you bury this material over time, you make coal, you make all of these organic things that we use, organic-based fossil fuels that we use, right? And so that's essentially was what the beginning of organic geochemistry was. Understanding where these molecules are linked to the principal formation of methane, hydrocarbons, and so on, crude oil, gas. Okay, so very important in terms of the origin of certain aspects of fundamental chemistry. I showed this the other day. I always have to show this because it's fundamental to the way we look at things. If you look at this pyramid, starting out with all of the organic matter in a water column sample, or all of the organic matter in a sediment sample, you can look at the carbon and nitrogen content of it. You can get elemental information about that material. You can look at stable isotopes. And then you can start looking at even more detailed things. You can look at NMR, you can look at biomarkers, and then you can look at compound-specific isotopes. As you go further down, you're decreasing the amount of total carbon that you're looking at in a sample because you start looking just at a particular molecule of a biomarker that's less representative of the whole pool of carbon. If you look at down here, you're also getting increasing source specificity. So while you lose the amount of carbon that you're looking at that's representative, if you're just looking at chlorophyll A or lignin or amino acids, you're looking at much less of the total pool, but it's much more specific, okay? So, and obviously the reverse going up. Okay, so a little bit, that was sort of the general intro on carbon cycling and some of these new fields of chemistry that came about in looking at biomarkers. This is a complicated slide, and you look at essentially these tree of life in the domains of eukaryotes, bacteria, archaea, right? So as you start looking at all these molecules here, some of these would be of interest to you, some of them may not be. If you go up, basically the bacteria, you can see that there are certain characteristic compounds. Chlorophyll A is in bacteria. You also have a number of methyl alkanes that are in there. We're not going to go through all of these, but you can see that if you take the tree of life and you start placing in key molecules, you can see that there are certain biomarkers that we have to track them. So you can look at okinones to track purple sulfur bacteria. You can look at green sulfur bacteria looking at some of these compounds, bacteria chlorophyll A, and so on. I won't take you through all this, but it's really fascinating to think about molecules that are unique to a particular plant or animal that we can then use to track over time, and once we've lost the ability to look at it microscopically. And all of these compounds have different decay stability. So for example, if you look at, let's see if I want to sort of take you through how much of this. So this would be trees and diatoms, conifers, let's say diatoms. And again, I don't want to take you through all of the specifics, but as you start getting into late stage decomposition, these compounds get more and more stable. Because what happens is the microbes start eating these molecules in the surface sediments, and then they start changing. So the molecules that are in the animals and the plants at this stage, living, as they sort of make their way down into these areas here, B, C, and D, going from 1 to 2 to 3 to 4, 5, 6, 7, 8, these are going into more, you lose the OH group. You start changing things. This compound is much more reactive to being broken down by a microbe than this compound. You can see you start stripping off a lot of the functional groups, and then you end up with compounds that are in the deep sediments that are still reflective of where they may have come from and are just more stable. Again, this is diagenesis, and this is the essence of how we use these biomarkers. Let's see if there's one more. Okay, beta carotene, that's what makes carrots orange. If you go into drugstores these days, you'll find lots of the things, lots of the pigments that people now take for longevity, because you'll see, you might see things like zeaxanthin, you might see things like lutein, and many people buy these things because aging in our body is considered to be an accumulation of free radicals. That's what happens. We get more and more free radicals in our body, and they tend to oxidize and break our tissues down. Well, the reason why plants have these is because they deal with the sun. They want the sun. They want it to activate chlorophyll A, but they have to control it so it doesn't turn into a chain reaction that actually hurts them, where they're actually being hurt by UV. So you want the chlorophyll to capture the sunlight for energy, but then you have to have something, because they do generate free radicals. So just the way plants use these compounds, like beta carotene, lutein, and zeaxanthin, to quench free radicals, we are now using that to have greater longevity. So it's interesting in terms of seeing how you can take these biomarkers and relate them to some very simple organisms and what we're doing for aging studies. This is showing terrestrial higher plants here. What are some of the things we would use? Lignin, N-alkanes, phenols, terpenoids, sterols. Again, this is just showing you, not in the tree diagram, but this is showing you some of the key biomarkers we use across a landscape. And again, all of these biomarkers are things that sometimes they work, sometimes they don't. So it's very difficult work to do, and it's very expensive work to do with the instruments that I'll be showing you in just a little bit. These are some other compounds. There are primary and secondary compounds. So let's just perform photosynthesis, CO2, H2O. Through photosynthesis, we get sugars. Now, the dark colors are primary processes. The light colors are secondary. So the primary means you need to do that to stay alive. The secondary, not so much. So what are these secondary compounds? Some of these are actually cardiac glycosides. What is that? These are, for example, if you look at mangroves and you extract cardiac glycosides in mangrove leaves, you'll find that they're enriched with that. And it's because mangroves live in areas where things want to eat them, birds, insects, all of that. So these secondary compounds are commonly used as a defense to stop organisms from eating you, right? And there's this coevolutionary thing going on all the time. The plant will make a compound that stops aphids, some sort of insect, from eating it, and then the insect develops or evolves an enzyme to combat that can eat it. This is what happens also in hospitals with antibiotics. We make super organisms because they keep adapting to the different antibiotics over time. And so it's this kind of cat and mouse thing going back and forth. So we can use these compounds as well that are in the secondary part of metabolism as indicators of grazing or indicators of organisms feeding on things. There's lots of clever ways you can use these different biomarkers. This is just some compounds that we have: chlorophyll, carotene, and phaeophytin. This is just a decay compound of chlorophyll A. So why would we want to look at the decay of the pure compound chlorophyll A? First of all, notice what's different. See, the magnesium in the tetrapyrrole structure is missing. So phaeophytin A is where the center metal in that particular tetrapyrrole has been removed by grazing, by an organism's enzyme. So when we see a lot of phaeophytin A in the water column, we know that the zooplankton are feeding on the phytoplankton. Okay, it's literally a grazing biomarker. You see a lot of it in the fecal pellets of copepods. Okay, I tell my graduate students this all the time. To get the pure compounds ordered, if you want to sort of start doing pigments in your laboratory to look at phytoplankton, you can dial up a Danish company and you can spend a lot of money for them to send you all the standards, thousands of dollars, right? Chlorophyll A standard, chlorophyll B standard, purified. Well, they have that benefit now, and I tell them when I was their age, we used to have to make our own, and sometimes we'd go to the vegetable store, and tomatoes have lots of lutein. Corn has lots of zeaxanthin, and we made and purified our own compounds. So I sound like an old dinosaur talking to them, and they can't believe that I used to do that. And it sounds like an old person saying they used to walk to school with just one shoe on, and it was a long way, and make you feel bad for me. But they don't feel bad for me. They just say, order the pigments. So anyway, I get no mercy from that. But you can do remarkable things. Like I said, you would expect that this particular pepper, orange, this one, that's beta carotene, just like the carrot. So they're really loaded with certain compounds. Vegetables, maybe is not what oceanography folks want to see. You can also get this from organisms like kelp, diatoms. You can purify the material from a large amount of algae to make your own compounds too. It doesn't have to be sort of the vegetable market. Same here, terrestrial sources. These would be the dominant terrestrial sources, trees, lichens, flowers, you name it. This is what would be in the landscape that might be coming into the coastal ocean in terms of the dominant sources. But it's not just pure plants. We also get lots of soil, and the soil is a mixture of everything. It would be a mixture of everything that's growing in this area that's in the soil. So it's more complicated than just looking at what's in the dominant trees. One particular compound is lignin, right? This is connected by ether bonds, beta O4 bonds. And basically, these are phenylpropanoid units that are connected. There is no one lignin molecule. There is chlorophyll molecule. Chlorophyll A is a distinct molecule. But when you say, give me a lignin molecule, I want a pure bottle of lignin, there's no such thing, because these molecules are mixtures of different families of lignin. So it's not that simple when you get to these kinds of compounds. But what we do is we can break these compounds down, this macro lignin structure, into particular oxidation products. And then we can use these specific products to look at their ratio and say, that looks like it's from a pine tree, that looks like it's from a flowering plant, or that looks like it's from moss, something like that, or wetlands. It's complicated and it's a lot of hard work. One of the really interesting stories about lignin is, if you look back in time, geologic time, the first vascular plant, right? Vascular plant, it has vascular tissue, appeared about 425 million years ago. It was about this big, and this is what it looked like, called Cooksonia. Here it is in the fossil record, and this is the first one to have lignin. The whole world before this time had algae, no lignin, no terrestrial plants. So what did this do? Well, amazingly enough, if you look at geologic time going in this direction, you see that there's a period where the amount of oxygen in the atmosphere went up to about 35. The present atmosphere is about 20%. So back in time, we know that the oxygen in the atmosphere at that time, from bubbles stored in ice and various things, we can say that the oxygen was higher. So what caused this? It turns out that the other thing you see is you see lots of organic matter burial. So we're burying a lot of organic matter, burial of organic carbon, right? This is the units here, right around the time where we see the high oxygen. And then this is the evolution of flowering plants, pine trees, and some other simple ferns. So how does this story work out? It's one of my favorite stories. When these plants evolved the ability to have this biomarker, this lignin, why did terrestrial plants need lignin? If you try to stand up a kelp plant here next to me, it will fall to the ground and flop. So basically you need lignin in the tree to defy gravity. Gravity will pull it down, right? You need something structurally to hold the tree up. You also need, if you're going to grow 200 feet high, or 100 meters high, you need to find a way to get the nutrients from the soil up. You can't just do it by diffusion like kelp do in a deep water column. So here's this simple molecule that I love to use to track terrestrial organic matter that changed the world for a number of years. So there was a new molecule in town, and the microbes did not know how to deal with it. So this new lignin molecule, structural support for big trees and plants and shrubs, and there was no bacteria to break it down. So what happens if the bacteria aren't eating it, right? Basically what happens is the oxygen goes up because there's less respiration. Okay? Bacteria use oxygen. So the bacteria are not eating as much of the organic matter because of this new compound. They don't know how to break it down. This is also important with fossil fuels. When you think of the Carboniferous period, when we start making coal, all of these different things that get buried in earth over time, millions of years, there was a period where you had a great deal of carbon accumulation. This is over millions of years here. So less bacterial decay, more organic carbon burial. You get deep burial, you get more of the Carboniferous period. For those of you who know anything about fossil fuels, that's a big period when we made coal and lots of oil and so on. So this is all from one molecule that changed. Excuse me, this is all from one molecule that changed and slowed things down. So basically this work did, and I don't know how familiar you are, but essentially they did some biomarker work, and they also did some genetic work. And it's estimated that before the fungi came around, so fungi, mushrooms, are the only organisms that know how to break down lignin. So if you think of a forest and you see a tree on the forest floor, you see mushrooms growing in it. They're the only ones that figured out how to break the lignin molecule down. So it took 200 million years based on the fossil work that for these organisms to find a way to break down lignin. So what does this have to do with oceanography? This is part of what I like to tell the oceanography people. Say, no, just start telling me about algae, just start telling me about phytoplankton blooms. I said, no. This is sort of the deeper, broader understanding of what biomarkers are, how they relate to broader concepts. And having that broader perspective will allow you to really understand things in a much more efficient way. So back to the limitations. I've given you sort of a very positive view on these pigments, or biomarkers, I should say. In this particular case, what are some of the problems? Here you have in situ production of phytoplankton. You can have algal coming in from the shoreline that you see growing on the rocks out here, right? All of that can bring in a suspended pool of algal material in the water. And then look at the shading here. So in here you have arrows that are hours to days, spotted here, speckled here. This is weeks to months. And then you can have seasons to millennia, right? So what happens is that as this algal material is eaten by phytoplankton, some of the phytoplankton digest it, they produce feces. You'll have chlorophyll broken down. The enzymes are breaking it down. So now you start getting this pigment that's moving through different pools of the trophic chain, right? Or the food chain. How reliable is it to now look in the sediments for something that you want to say over time? For example, if you look, the rivers coming into the Arabian Gulf are being impeded now with damming, less particles coming in from upstream, and you change the nutrient dynamics with respect to algae growing in and around. Maybe you get more harmful algal blooms with heat in parts of the Gulf. But what you also get is actually you change all of these dynamics. What's coming into the sediments, what gets stored in the fossil record. So to look at sort of a long-term change of what's been happening over the past 5 decades or 100 years as you start changing what's coming into the northern part of the Arabian Gulf, you would have to take a core with Mohammed's group, and we would go down, and then we can look for certain biomarkers to say, it looks like diatoms were here, dominated, now this has changed to dinoflagellates and so on. You can see the historical changes over time with biomarkers. Or can you? Sometimes it's not very good. Sometimes the preservation is not very good. If you get too much of this breakdown and oxidation, saturation, rearrangements of the molecules, it might be very difficult to describe or reinterpret what's happened, the reconstruction of what's historically happened in terms of riverine inputs and phytoplankton in the northern Arabian Gulf. It might be difficult to see that, or maybe not, depending upon how well it's preserved. This is the kind of thing that we see in sediments. So this is what I'm talking about. You have the plankton up in the water, then you have a trap, sediment trap at 105 meters, a sediment trap at 1,000 meters. So let's just stop right there, and then 1 to 3,500. So let's just stop right there, right? So this is essentially what's traveled from the surface ocean down to 3,500 meters. And what you start to see is that we have certain classes of compounds. Amino acids, you can't see the difference between amino acids and sugars. The sugars are a little bit lighter here if you see. I apologize for that. And the amino acids would be over here, and the lipids. And then we have this huge pool over here, uncharacterized. That's the biomarkers person's nightmare. That's the material we have no idea what it is. So we can't separate everything into something that's nice and clean, lipids, pigments. We can't do that. Nature has created things that are outside of our analytical window. We can try the best instruments we have. And now this was 1997, Stu Wakeham. This has gotten smaller in terms of what we can actually see and breaking it down. But you can see the point is that you can't use the biomarkers all the time. It depends on the system. And then when you get to the sediments, just in the surface sediments or 10 to 12 centimeters down, you see even more loss. So you can see the selective loss of material, and the lipids are more stable than the amino acids and the sugars. So they change more quickly from the surface to the bottom, depending upon their structure. Okay. This is transformation of biomarkers with increasing maturity. So as you go deeper, you start to get decay, right? This is essentially pristane, hopane, steranes, sterols. All I want you to sort of get from this is that you start to see the production of certain compounds with depth. So these are very stable compounds. You might say they're actually increasing with depth, increasing maturity. Why would something increase with depth if I'm telling you that once it lands in the sediment and once the microbes start chewing on it, it becomes a different molecule, or it's totally lost and converted to CO2. So this particular diagram here is showing you certain compounds that become more prevalent because of the inability of microbes to break them down or become byproducts of an original compound that was in the living cell. So these are very stable compounds that were converted from other molecules into these, and they actually increase with increasing maturity. So when we see these compounds, we know that these sediments have been there for a long time and that we're looking at biomarkers that reflect long-term degradation. And some of them still give us where they're from, but it's mostly used as a decay indices, right? The decomposition indices. And this is another sort of schematic that looks at the microbial record in the geosphere. And there are different ways that you can look for microbes. So there are two types of microbes. We have the archaea, right? And then we have the eubacteria, right? So these are, of course, you can use RNA and DNA. RNA has a very, very short lifetime, right? Not very stable. DNA you can get over thousands of years. So that's genomic. And then you have these other compounds, molecular. This is intact phospholipids, and polar lipids. I can say intact polar lipids, I would say. I'll take that back. And then polar lipids and hydrocarbons. So again, the intact polar lipids, very short lifetime, and some of these other ones longer, and so on. So, and the morphology of the cells, you can maybe compare with these particular compounds, just looking for how the microbes have been changing in the fossil record, or in the sediment record. Another one here shows you some other biomarkers, and I won't bore you with the names of all of these things. But the point is, you can see that some biomarkers are very useful over tens of thousands of years, as I just showed you a minute ago, and some of them are much more restricted in tens of years. The other thing that's important is that this tells you also not how long they've been there, but there are certain particular compounds that reflect whether they've been in an oxygenated environment or they've been in an environment with no oxygen at all. So this can also start to give information about whether the water has gone anoxic or has stayed oxic? And that's a question that might be interesting in the Gulf here, where you're starting to see more and more low oxygen water as it moves out from this lower center part of the Arabian Gulf and how it may interact with the Arabian Sea as it moves out, or you get this exchange of water. But some of these compounds might be very useful to look at changes in the water column oxygenation or not. Or redox changes, what we call paleoredox, right? You're interpreting the oxygen record over time. Another thing that I could just sort of mention to you, this was work that we did in New Zealand, in Fiordland, New Zealand, and we can start to actually look at first order decay rates. That is a decay rate of per year. It has a K, decay rate, 0.004 to 0.003. So you can start calculating these biomarkers based on their changes from the surface to the deep. At the rate that they're changing, you can start calculating a distinct decay rate. You might say, who cares? Well, again, to try to figure out the magnitude and the dynamics of how quickly things are decomposing, once you can put sort of a timeline on it, that's very useful. But we cannot do that without radionuclides. So the radionuclides, beryllium-7, thorium-234, lead-210, if you take the biomarker data and you combine that with what we know about the sediments accumulating or eroding, you can start to put a picture together. We know the sediments are accumulating this fast, or we know they're being exported. And now let's look at the biomarkers. We know that this decay rate would definitely fit. Yeah, most of that chlorophyll A is gone within the time frame of the mixing based on beryllium or thorium. So this is the way you start piecing the different puzzles together to look at how quickly material is being decomposed or not. Now, this was from Harry Elderfield. He was a well-known trace metal radionuclide person in Cambridge University. And this is the way he typically described listening to biomarker people talk. So this is proxy confidence factor, right? So when we discover a new biomarker, we get up to the euphoria phase. We're very happy. We know it's going to be there. We know it's very specific to a particular algal cell. And we're all happy we've got a new biomarker for dinoflagellates that is going to be the best biomarker we've ever had. Then you realize that what happens is you start to go through this crushing phase because someone now found that biomarker in 10 other different types of algae, or it's not very stable. So then you go from the happy phase to this horrible phase of pessimism and say, this is never going to work, and more people are discovering that it's not very specific. And then eventually you get into this sort of realism phase where you can say, yeah, I guess we can do this, but we can't really say it to that degree. And so you wind up sort of midway up this between euphoria and pessimism. And I would say that this happens many, many times with biomarkers. We're always looking for better ones, and we always go through this phase, unfortunately. So now we can go to this compound-specific approach to things. I was mentioning this to you the other day, that we can look at specific stable isotopes and radiocarbon age of particular molecules. That takes a lot of work. You can collect this from an algal material. You can put it through a column and separate it to different types of compounds. So let's say now we have brassicasterol, if this compound is indicative of diatoms. And we only want to look at just that one compound in the sediment because we know then we're only looking at the age of the diatoms as we go down core. If you don't do that and you just look at radiocarbon and stable isotopes, you're looking at the age and the sources of thousands of different carbons. This is telling you specifically what's happening with a particular source. So that's why they call it compound specific, and then you can sort of isolate material and do radiocarbon or whatever. So that came about in the 2000 or so. It became more popular. These are just some of the instruments I have in my laboratory. This is the instrument that will separate a particular compound, a particular amino acid, a particular lipid, and it will give you, it will automatically inject for you 1,000 times, let's say, to isolate that compound and then get it isolated in these little tubes here that have liquid nitrogen in them. And then you wind up getting a very small amount of material that is a pure compound that you're interested in, that you then have your own standard that's been collected. And you can then take that and not so much use it as a standard, but you would want to know the age and the stable isotopic composition in where you collected it. Let's see, this is another, this is our stable isotope where we can measure C13, N15, so on. This is a gas chromatography triple quad system. That means GCMS. And this is a liquid chromatography system, triple quad MS. All of these things are really annoying to work with. They like to break, they're really expensive, they give me gray hair, and they just drive me crazy. But when they're working, it's so nice and wonderful. I talk to them very nice. The students don't see me doing that, but I know it makes a difference. And then finally, I just have 2 more slides and we're finished. Basically, another approach, it's a little, not really biomarker per se, but these are these big instruments that cost about $3 million. And these are things that we basically refer to Fourier transform ion cyclotron resonance mass spectroscopy. And what you can do in this is you can get thousands and thousands of molecular formula. So you inject it into this machine, and you get all of the organic formula, every molecule that's in there. And then you have to figure out a way to sort that out. And what we do is we use what we call these Van Crevelen diagrams. You know that there's a certain hydrogen to carbon ratio of all the molecules in there, right? And you know that the organic carbon or oxygen to carbon ratio. And then you can separate based on what we know about true lipids, true proteins, amino acids. You can get all of these particular thousands and thousands of molecules that are coming out of your one sediment sample or one phytoplankton sample. And it's not specifically like the work I'm doing where you have a particular biomarker. This is everything, every molecule that's in there, but we can't quantify them. What we can do is just more of a qualitative way of looking at, and I can't get into why you can't do that, but it has to do, well, anyway, basically you're limited by just sort of looking at how much of the lignin and where it's sort of focused. The final thing is, so that was FTICR, which we call high resolution mass spec. This is the way you would measure radiocarbon. If you have a sample and you want to know how many thousands of years it is or whatever, this is the way radiocarbon used to be measured. It still is to some degree with a very large AMS. And most of the time you have to send it to a particular lab. This is not the kind of thing you have in your own lab. There's a facility that everyone runs and you send it to them and it's about $400 or $350 a sample. And that's why people, when they look at down core and they say, why did you just do 2 or 3 radiocarbons? Why didn't you do many more? And you realize how expensive it is. So I was at Yale University on a scholarship last semester. Old people still get scholarships, believe it or not. And this is the new system. It's called a Mercatus system. This is Pete Raymond, my friend. It's at his facility. So this is considered to be now the little brother of the one I just showed you, that you can measure radiocarbon. And you can measure as many radiocarbon as you like. You don't have to worry about sending and just saying, give me 3 samples or 4 samples, I can't afford more. They go for about $1 million, and this is what people are doing now. So the whole idea of radiocarbon, whether you do compound specific or bulk, this is now considered to be the small tabletop. It's still a bit of a beast, but compared to what I just showed you in the previous slide, it's considered to be very small. It was invented in Zurich. I met the genius who invented it. I was visiting the people there, and I said, who's that strange gentleman over there, and they said, that's the genius that invented it. And I went over and chatted with him a bit when I was in Zurich at ETH University. So I got to meet them in Zurich, and it was pretty amazing. Now they're millionaires because people are ordering these things all over the world, not having to go to these big facilities. So that's a little bit on the sort of end point of stable isotopes and radiocarbon, and I'll stop there. Thank you.
Identify species.
Oh, I think that they're already involved in that, yeah.
Okay.
So the whole idea of morphology and the whole paradigm of identifying things, phylogenetics, is something that has changed dramatically over the years. Not just because of biomarkers, but even whether you can use the appearance of an organism as truly reflective of a speciation event. Because there are many people now that see that if you're just using what it looks like, right, you can be misled, because there can be genetic changes that don't result in a physical change in the feature, enough to make it a different species. And that was really a big problem. I don't know if you follow what I mean. That phylogenetics was a huge thing. They've been arguing for years about whether this species diverged or it didn't. And you can't just use the phenotype, you have to use the genotype. So the biomarkers fit into that a little bit better, if you can get that.
Right. Thank you. I do have another question that might sound a wee bit silly.
No.
But why did photosynthetic organisms decide to stand up when they could have just laid down and didn't have?
Yeah, that I'm not sure I can answer. I mean, the question is, why would vascular plants, when everything is sort of soft and mushy and algae and small and not big structural trees, why would plants want to start growing away from the soil? They are going towards the sun, but you can still get plenty of sun. I have no idea why that occurred, why Cooksonia, this little plant that now had this magic molecule in it to grow up to be hundreds of meters high. I'm not sure what the driver was, but it would have to be some sort of selective advantage for those species to pass on those genes. And I don't know what that selective advantage would be. I mean, it gave them structural support, but as you say, if you can live without that, why would you need the structural support? So yeah, it's a good question. I don't know.
Just as a comment to that, I think it's just like your barnacles. It's really a real estate issue. And if you can spread out, it's like having a high rise when land is really expensive rather than having a one story. I think it gives them a competitive advantage to go vertical.
I think that's a good point.
There's no space.
Yeah, you're not competing for space laterally. You just go up and create a canopy where you gather the sun and let all the other short guys fight with each other down below for space, and you're up above. So yeah, exactly.
Are there specific databases to see, let's say if I locate certain biomarkers in sediment, how accurate could I trace it to a specific species of pine tree in Russia? Like, does it go that far?
We never really know how far we're getting. That's a good question. This is why we always have to use what we call end members. So if you go and you look for lignin in a lake in northern Russia, and you haven't at least sampled some of the dominant trees and extracted and looked for the biomarkers, you would think, look at the lignins, look at the chlorophylls, look at the carotenoids. You always have to have your biomarkers to help you figure out what percentage is going on. So we can't just say someone measured some soil or someone measured this pine tree in Siberia and I'm going to use that. You have to use it around that specific area. So we would go and we would find local sources that would contribute and then develop our own library and then do mixing models that figure out, based on the end members, what percentage is pine, what percentage is maple, what percentage is oak, based on the dominance. You can't get all of the end members. You get the dominant ones. So it's a lot of work to do. I mean, that's a good question. I didn't really mention that you don't just go and start collecting sediments and water and ignore the surrounding area if you're interested in terrestrial inputs. And it depends on so to answer the final question, does it work down to the species level? Usually not. If you're talking about different species of pine trees.
Okay.
That's really, really getting beyond the limits.
More question first? I think you have another question.
Yeah, because I'm thinking about the practical use. Like the diatoms, for instance, you could see specific biomarkers from diatoms. Could I see something that came from the Pacific in the Gulf necessarily? Like, could I see, okay, this is a diatom from the Pacific. Maybe it's come from ballast water.
That's going to be a tough one. That's going to be a tough one because the diatoms generally have a similar makeup. When you start to see differences in certain diatoms where they live, if they live in different parts of the water column, then you'll start seeing different ratios of the carotenoids because certain carotenoids absorb certain types of light. So the deeper you go, the more you get the red light washed out and the more you get the penetration of the higher photon energy blue light. And sure enough, the ones that grow deeper, if you look at their the peaks, the absorption peaks, when you look at the molecules, they're absorbing in that lower UV range, right? I mean, it's really amazing to see how they've adjusted to light and what light looks like at 10 meters versus light at 50 meters. So they're living there and they now have the molecules adapted to absorb different photon energies.
More questions from students? I'm sure some of you are familiar with lignin, yes? Reem? Yeah.
Good questions from you guys.
More questions? So, thank you, Dr. Bianchi, for this wonderful talk.
Uh-oh.
Thank you so much. It was a privilege and honor to have Professor Tom Bianchi with us. I think his talk was very informative and he provided lots of nice science
So this is
answers to us.
Oh,
this is the boom,
oh no.
and yeah. and
Wow.
You remember the guy that I showed you the first day? That
Yeah.
He designed the boom. His
Yes.
name is Ibrahim Felkawi. So he's the one who designed this.
Oh, wow.
And there's
That is so nice.
This side in English.
That's
and
beautiful.
It's in Arabic here.
Wow, that's
Dr.
fantastic.
Thomas Bianchi. This is in Arabic.
Wow.
So this is a handmade and.
I'm very happy.
Small
Just-
token of appreciation.
Okay.
Oh, you can hold it like this.
Okay. Thank you so much.
Thank
Oh,
you for
very
visiting
generous,
here.
very generous. Thank you so much.
It's a pleasure. Thank you so much.
Okay, yeah, sure,
Maybe I'll put it for the Arabic side.
yeah. As I mentioned to some of the students before, if any of you are sort of interested in some of the things, I talked about a lot of different things in the two talks, and I know it was sort of full of a lot of material, so I realize there's no way you can gather all that information that quickly if you don't work on this in these particular areas. But feel free to email me about anything. You know, just say, I saw your talk and I'm reading this and I'm just curious about this. I'll respond. So just feel free to do that. I'm happy to interact with you on that.